Assalamu alaikum everyone, this is Abdullah Khalil and this is the platform where you can learn O-level, IGCSE and A-level biology of different UK boards. The first topic that we are going to do today is cell structure because it is the basic topic of biology. Now this topic is from AS level biology 9700, the board is Cambridge Assessment International Education. Whenever we are learning the topic in biology or in any other subject, the first and foremost is to look at the learning objectives provided by the board. So these are some of the learning objectives that you can see. We will skim through this and then we will move on to the first topic. So the board says that you should be able to make temporary preparations of cells and you should be able to view them under the light microscope. So if we have to observe the cells, we are going to make a preparation, a slide, which is going to be observed under the light microscope. So we can observe animal cells and plant cells with this method. The animal cells generally that we use are the skin cells or the cheek cells, and the best specimen for the plant cell would be the onion epidermis. Then we should be able to draw the cells from the slides or photomicrographs. Photomicrograph is again the picture from a light microscope. If it is taken from the electron microscope, it would be electron micrograph. Calculating magnification, actual size of drawings or photomicrographs or electron micrographs, and using the eyepiece. Eyepiece is one of the important part of light microscope. We will go through the structure of or the diagram of light microscope as well. Another important thing, conversion of units for measurement. Now when we are calculating magnification, the actual size or the image size, it is really important that we are able to convert the units, because the two units that are provided for actual size and image size, let's say, if the actual size is provided in micrometer and the image size is in millimeters, when you're calculating the magnification, you need to have both of these in the same units. So you will be converting units. You should be able to convert centimeter into millimeter, millimeter into micrometer, micrometer into nanometer. So centimeter till nanometer, these are most important units for microscopy. Magnification and resolution, two of the very important concepts of microscopy. You should be able to know the difference between them. You should be able to explain what a resolution is for a microscope and what magnification is and how they're different. In the next part, cells as the basic unit of living organisms. These are some of the learning objectives in front of you. Recognizing cell structures and organelles. Now you should be able to recognize the cell structure. That means you should be able to know, you should be able to identify if it is a bacterial cell or a plant cell or an animal cell. Okay. Obviously the specialized cells are also there, so you should be able to see if it is the red blood cell or a white blood cell or a root hair cell or a xylem vessel or any other different type of plant cell or animal cell. The next is to understand their structure and function. Now this is about the organelles. In eukaryotic cells, you're going to see a lot of organelles. You see mitochondria, Golgi bodies, chloroplasts, endoplasmic reticulum, nucleus, ribosomes. So these structures are really important. You should be able to identify, label and name these structures. Plus you should be able to relate their structure with their function. How is mitochondria, how the structure of mitochondria is related to its function, how the structure of chloroplast enables the chloroplast to help the plant in photosynthesis. So you should be able to do that, and we will go through it briefly here and in some other topics as well. Key structural features, describe, compare and interpret photomicrographs, electron micrographs and drawings of typical animal cell and plant cell. So if you're given a typical animal cell, obviously there is no such thing as typical animal cell and typical plant cell. All the cells are very different from each other, but the generalized cell that we study in our book. So if you're given a plant cell and an animal cell, how are you going to compare them? What are the key differences between a plant cell and animal cell? How are you going to identify that? And what are the similarities between plant cell and animal cell? Then key structural features of prokaryotes as in bacterium. When we are studying prokaryotes, now we know that fundamentally there are two types of cells. There is prokaryotic cell and there is eukaryotic cell. So prokaryotic cell is the one which is very old, billions of years old cell from which all the life forms have been evolved. This is the cell which does not have the nucleus. That's the major difference between prokaryotes and eukaryotes. If you're asked to differentiate between the prokaryotic cell and eukaryotic cell, the first thing that you want to say is that the prokaryotic cell does not have a membrane bounded nucleus. However, it has the DNA, but the difference is the DNA is the naked DNA, it is scattered in the cytoplasm. There is no membrane bounded covering for that DNA. While the eukaryotic DNA, first of all, it is present in the nucleus, which is a double membrane bounded organelle. Secondly, this DNA is not just DNA. It is integrated within the chromosome. So there are histone proteins in which the DNA is wrapped, and this structure is called the chromosome. Then we have the structure of viruses. This is the last learning objective. Now let's move on to the first topic of this chapter, making temporary slides and drawing cells. Now there are two types of slides. There are prepared slides, which generally contain the dead cells. They have been preserved in life-like form, but they're not actually living. What we make in the lab is actually the temporary slides, in which we can observe the living cells. Now the living cells, as I've told you before, the living cells that we observe are the plant cells and the animal cells. In plant cells, the best specimen is the onion epidermis. However, these cells are transparent, so we need to use stains so we can observe them under the light microscope. So the stains that we use for the animal cell is generally methylene blue, and for the plant cells, generally we use the potassium iodide in iodine solution. Here we have preparing a temporary slide. For this, you can also use chopped up material. For example, if you're observing xylem vessels, you will be using chopped up material. The stains that we use, already told you, iodine and potassium iodide solution for plant cells like onion epidermis, and methylene blue for observing animal cell like for the cheek cell and for the skin cell, whatever we are observing. This is the basic method in which we are going to prepare the temporary slide. Now slide is this glass structure, and the first thing that you do, for example, let's say we are observing the onion epidermis. You cut the onion, you take out a small piece, small skin of onion using a forceps. This is your specimen. You keep your specimen. Now there is the slide that you have to prepare. With the slide, the first thing that you do is you place a drop of water so the specimen can stick to it. Then you place the specimen on top of the water, which was in this case the onion epidermis. Now here we are lowering the cover slip. But before lowering the cover slip, there is one thing that we have to do here, staining. So we are going to stain it with, because it is onion epidermis, because it is a plant cell, we know that we will be using potassium iodide in iodine solution, which is also called iodine solution for short. So we will be using iodine solution for that. Then we are going to lower the cover slip gently and smoothly so there is no air bubble trapped inside. Because if there is an air bubble trapped, it would obstruct your vision. This is how you prepare the slide. Now this slide is ready. You're going to give it a few minutes so everything is dry. You can use the filter paper to soak up the extra water or the extra stain, and then the slide is ready to be viewed. Now you can place it on the stage of the light microscope. Let's move on to the next topic. The basics of this that we have studied in our IGCSE O-level or high school. So we already know all the living organisms in the world are made up of cells, not viruses, because they are not considered as living organisms. Secondly, cell is the basic structural and functional unit of life. Fundamentally, there are two types of cell. There is prokaryotic cell and eukaryotic cell. I have briefly discussed it with you. The bacterial cell or the prokaryotic cell is the one that does not have the membrane bounded nucleus. It does not have the DNA integrated with the proteins. It's just the DNA. But in eukaryotic cell, the DNA is integrated with the proteins in the structures called chromosomes, and it is present in the membrane bounded nucleus. That is eukaryotic cell. Now, in eukaryotic cell, membrane-bounded organelles like mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, etc. May be present. But in prokaryotic cells, there are no membrane-bounded organelles. Even the ribosomes are different. The ribosomes in prokaryotic cells are 70s, slightly smaller in diameter. They are 29 nanometers. In eukaryotic cells, the ribosomes that are present in the cytoplasm or the ones that are attached to the endoplasmic reticulum, the rough endoplasmic reticulum. Those ribosomes are 25 nanometer, slightly larger in diameter. These are 80S ribosomes. But the surprise is that within the eukaryotic cell, there are organelles that may also contain 70S ribosomes, which we will discuss later. Also, the cell wall, bacterial cell has the cell wall, but it is made up of peptidoglycan. When we talk about eukaryotic cell, which eukaryotic cell are we talking? The animal cell does not have the cell wall. The plant cell, it does have a cell wall, but it is made up of cellulose. The fungal cell has the cell wall, it is of chitin. So different materials are there. Then cell and microscopy. Now when we are observing cells, obviously the observation of cell or the study of cell became possible because of the microscope. So let's try and understand how the microscope works. We have two types of microscopes. We have the light microscope and the electron microscope. The basic principle is the same. The basic principle is for any microscope to use radiation as the source, and the specimen basically obstructs the pathway of the radiation, and then this specimen can be viewed. So light microscope, as the name suggests, is using the light radiations, the light waves as the source. An electron microscope is going to use the electrons, because the free electrons can behave as electromagnetic radiation. So these radiations can be used as the source for electron microscope. First, let's study the light microscope, which is much simpler as compared to the electron microscope. You can see at the top we have the eyepiece, because we observe through it. Within the eyepiece there is a lens actually. So we have the eyepiece lens and we have the objective lens at the bottom. Objective lens is the one that is on top of the specimen. The specimen is present on this slide, which is on the stage. This stage actually has a small hole through which the light can pass through the, here we are using mirror, but in the modern microscopes we have the light source, a bulb here. The light from this bulb goes through the hole and then in the lens and through the eyepiece goes into our eyes, so we can observe this specimen. So on top of this, the specimen is placed in such a way that it comes on top of the hole, so the light can pass through this. Okay, so this is the labeling of the light microscope. When we observe an animal cell, as I said, the generalized animal cell, a typical animal cell, the word that we use is typical, but there is no typical animal cell actually. Every cell has a different structure. But the generalized animal cell would actually be seen like this when observed through the light microscope. Later we are going to see how the animal cell would appear under an electron microscope, and you should be able to know the differences when the same cell is observed under the light microscope and when it is observed under the electron microscope. Now this is how the plant cell would appear under the light microscope. Again, we are going to see how the plant cell would appear under an electron microscope. So make sure you're able to identify the differences. This is the plant cell as seen under the light microscope. We can see different structures which are labeled here. The nucleus, within the nucleus we have the nucleolus and the chromatin, the Golgi bodies, chloroplast, mitochondria, cell wall, cell membrane, plasmodesma, which is the connection of the cytoplasms between the neighboring cells of plants. Now we have the electron microscope, much bigger than the light microscope, and the working is very different. The basic principle is the same. Now, to understand why we need electron microscope when we already have light microscope, see, the limit of resolution, this is the rule. The limit of resolution is generally around half of the wavelength. Half of the wavelength for light microscope, if you know the radiation, the wavelengths of radiation, the visible light expands from 400 nanometer to 700 nanometer in wavelength. The smallest being the 400 nanometer. So let's take 400 nanometer as the wavelength of light. What is the half of 400 nanometer? 200 nanometer. So anything that is 200 nanometer or above can actually be observed with the light microscope. And those are some of the best refined quality light microscopes that will be used to observe something which has diameter of above 200 nanometer. Now, a while ago we discussed about ribosomes. The eukaryotic cell has ribosomes which are 25 nanometer in diameters. Can you observe ribosomes with light microscope? No, because ribosomes are much smaller than 200 nanometer. So what is the solution? To use some radiation which has much smaller wavelength, so we can observe even smaller structures. After this, we are going to compare magnification and resolution, but for now, just comparing the electron microscope and the light microscope. So electrons are used as electromagnetic. There are many other options as well, but why are we using electrons? Because they are charged particles. They can easily be focused. We need to focus the electrons on one plate so we can see the image. So electrons are charged particles. They can be focused on one plate. They can move and behave like electromagnetic radiation, and their wavelength is much smaller than the light. So when we use electrons, they are negatively charged. They can be focused using electromagnets, and the resolution that we obtain is 0.5 nanometer. Now compare this with the light microscope. The best resolution with the light microscope is 200 nanometer. With electron microscope, 0.5 nanometer. So ribosomes or even much smaller structures than the ribosomes can be observed using the electron microscope. Now, this is how the animal cell would appear if we observe it under the electron microscope. If for a second we go back to the structure of animal cell that we observed with the light microscope, you can see so many differences. There are so many extra structures that you can see with electron microscopes. For example, the ribosomes. Even the mitochondria would appear very different because you can actually see what is present inside the mitochondria. Though they were observable with the light microscope, but you couldn't see what is present inside the mitochondria. So this is how the animal cell would appear under the electron microscope, and this is how a plant cell would appear under the electron microscope. The main difference is ribosomes. You cannot see ribosomes at all with light microscope, and you can see ribosomes with electron microscope. Then comes the magnification. Obviously, there are questions given to you where you calculate magnification. Whenever you are asked to calculate magnification, there is the actual size. The actual size of the bacterium, let's say, is 3 micrometer. You observe it with the microscope, and the new size you get is much larger than that, let's say 5 millimeter. Now you're going to calculate the magnification. This is the formula for magnification that we are going to use. Now this is a triangle, so even if one value is to be calculated and the other two values are given, we can do that using the same formula. So even if we have to calculate the image size while we have the magnification and the actual size, we can do that using the same formula. Or if we have the actual size and we have to calculate the image size and magnification, we can again do that using the same formula. The formula changes as per the need. Now there is an object, let's say this is the object, this was 0.3 millimeter in diameter. When observed through the microscope, it becomes 3 centimeter. The first thing that you're going to do is you're going to use the formula, put the values, then convert the units. Do not forget to convert the units, because one of the units that we are getting here is millimeter and the other one is centimeter. So they should be in the same units. We will convert centimeters into millimeters. We know that 1 centimeter has 10 millimeters, so 3 centimeter would be equal to 30 millimeters. 30 millimeters divided by 0.3, so that means the magnification is 100. You can do different questions using the same technique. Resolution, how it is different. Now magnification is how enhanced, how large the object appears from its actual size. This formula, this calculation tells us that the object has appeared 100 times larger than its actual size. Resolution is not about that. Resolution is how clear the picture is. Okay, it is By definition, resolution is the ability of a microscope to distinguish between 2 points, 2 objects that are closely lying with each other. So the ability of a microscope to distinguish these objects is resolution. And there is a point, as we said, for light microscope, 200 nanometers. After 200 nanometers, if the 2 objects have distance less than 200 nanometer, the light microscope would not be able to distinguish those as 2 separate objects. So this is what resolution is. Higher resolution would give you the clearer picture. Even let's say you have a picture with electron microscope and with light microscope at the same magnification. But even these 2 pictures, the electron micrograph and the light micrograph, would appear very different from each other. The electron micrograph would be much sharper and clearer because it is at high resolution. Now, this is the depiction of how light microscope and electron microscope works. When we are using the light microscope, because the wavelength is much larger, the larger objects like mitochondria here can obstruct the pathway of light microscope, of the light rays, sorry. So if the light rays are moving in this direction and the mitochondria obstructs or blocks the path of light rays, this mitochondria can be observed using this light microscope. But the smaller structures like ribosomes here, as you can see at the bottom, they cannot be observed with this light microscope because they're very small to obstruct the pathway. They are very small, way too small to block the pathway of light rays. When we use the electrons, they have much smaller wavelength as seen here. Now these wavelengths or these rays can be blocked by the mitochondria as well as ribosomes, or even smaller structures can also block the pathway. So they can be observed using this. Now, electron microscope is also divided into 2 types, or there are 2 types of electron microscopes that we generally use or see, or the electron micrographs that are given are generally from these 2 microscopes. There is the transmission electron microscope and there is scanning electron microscope. Now, you can learn this using the name. The name suggests that the scanning is the one that scans. The outer structures can be seen. When you have to observe the outer structure of something, the surface of something, you will be using the scanning electron microscope. But let's say you have to see within that cell, within that structure, inside the structure. Now you're going to use transmission. So the transmission electrons are the ones that can transmit through some object. Scanning are the ones that are bouncing off the surface. So you can observe the surface using the scanning electron microscope, and you can observe the internal structures of something using the transmission electron microscope. Another important calculation which was there in 2024 paper for AS is the eyepiece graticule. Now I don't remember which variant was that, but one of my students has given me the feedback that there was a question about eyepiece graticule. It is placed inside the eyepiece. What is the graticule? It's a small scale. It's a small transparent scale which is placed within the eyepiece. So when you're observing something, you will be seeing the specimen as well as the scale. This is used to measure something. Let's say we are observing cells and you want to measure the diameter of a cell. So you will be using this eyepiece graticule and you will see an image like this. You can see the scale as well as the cells. As per the observation, we can see the cell that we are measuring here, it appears between 40 and 60 of the eyepiece graticule divisions. That means what is the size of the cell? 60 minus 40, 20 divisions. 20 divisions of eyepiece graticule. But what is the size of one eyepiece graticule division? We don't know that. So actually, to know the size, the next thing that we are going to do is to calibrate it. So how do we calibrate it? There is another scale that we are going to use. Now, because this is called stage micrometer, obviously it is placed on the stage. In the light microscope, we have seen where the stage is. The stage is the area where we place the slide generally, where there was a hole. So that's the stage. At the stage, we place this stage micrometer. In the eyepiece, we have the eyepiece graticule. Now when we observe, we are going to see both the scales. We are going to bring it into focus in a way that both the scales are aligned like this, as you can see here. So when both the scales are aligned, the stage micrometer has the known divisions. So we have 0.001 and 0.01 and 0.1 divisions here, 4 millimeters. The total size is 0.25 millimeter. For this stage micrometer, the total size of this stage micrometer is 0.25, which is equivalent to 100 eyepiece graticule divisions. When we calibrate this, we are going to divide 0.25 by 100, so it will be 0.0025 millimeter. That's one eyepiece graticule division. And when we are going to convert it into micrometers, it will become 2.5 micrometer. So that means one eyepiece graticule division is equivalent to 2.5 micrometer. What was the size of this? 20 divisions. One division is equal to 2.5 micrometer. We are going to multiply it. So 2.5 multiplied by 20, that means 50 micrometer. The size of that cell is 50 micrometer. Once the eyepiece graticule has been calibrated, now we can use it again and again. We don't have to calibrate it every time. Now the conversion of the units. As I said, the most important ones are centimeter, millimeter, micrometer, and nanometer for microscopy. But let's start with kilometers. Yeah. 1 kilometer, we know that 1 kilometer is equivalent to 1000 meters. 1 meter is equal to 100 centimeters. 1 centimeter is 10 millimeters, as we can see here. And finally, we have 1 millimeter, which is equivalent to 1000 micrometer, and 1 micrometer is equal to 1000 nanometer. So if you remember this conversion, or you can do it in obviously in the exponential form, right? Like for example, 1 micrometer is equal to 10 to the power minus 3 nanometer. That would give you the same result. Whenever we are going from the bigger unit, like centimeter is the bigger unit, I have mentioned here, on the left side we have the bigger units. So whenever we are going from the bigger unit to the smaller unit, what we will do? We will multiply. When we are going to this from the smaller unit to the bigger unit, like if we are going from meters to kilometers or nanometer to micrometer, smaller unit to bigger unit, we are going to multiply.